Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com and Cactus webinar titled Discovery and Engineering of High-Affinity Soluble T-Cell Receptors via a High-Throughput Yeast-Based Platform. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. Garrett Rapazzo, Senior Scientist in Platform Technologies at Adimab, who will present his team's research on the discovery and engineering of soluble T-cell receptors for the use in potent and specific peptide-HLA targeting therapeutics. All right, so with that, I'm very pleased to welcome Dr. Garrett Rapazzo. So Garrett, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thanks, Liam, and thanks everyone for joining the webinar. My name is Garrett Rapazzo. I'm a senior scientist at Adimab. Today I'll be presenting on the discovery and engineering of high-affinity soluble TCRs via a yeast-based platform. So to give you an overview of the talk, I'll first be presenting on the background of soluble TCRs, TCR mimetics, and in general peptide-HLA targeting therapeutics, going into the design and validation of our soluble TCR platform in yeast, and then into an affinity maturation pilot that we did with a model TCR. Next, I'll be presenting on our naive discovery capabilities with soluble TCR libraries, and finally, a summary of the presented work. So first, a background on Adimab. So we are a company of protein engineers that work with small biotech partners through large pharma partners to bring their therapeutic programs into the clinic. And we've been at this for a long time in the antibody space, but we've recently gotten into the space of both TCR mimetic antibodies as well as soluble TCRs for both engineering and discovery. And the reason we've gotten into this space is that relative to traditional antibody therapeutics, which can only target proteins either secreted from the cell or on the surface of the cell, targeting peptide-HLAs gives us the ability to target the intracellular space of a cell, opening the target space much more broadly. These include proteins like oncoproteins or viral proteins that are only accessible through the HLA presentation pathway. And these are then recognized by T cells on the surface of the cell and can be co-opted by things like soluble TCR-based therapeutics and TCR-based mimetic antibodies. Comparing these two modalities, TCR mimetic antibodies and soluble TCRs, you can see that TCR mimetic antibodies are very well studied and have a lot of data on developability properties for antibodies in the clinic. They're also highly stable with few to no glycans, and by contrast, have no evolutionary basis for peptide-HLA recognition. This means that antibodies will target many different epitopes within the peptide-HLA, and therefore might not be inherently peptide-specific. However, TCR-based therapeutics have an evolutionary ingrained peptide-HLA specificity that allows them to be safer for use in therapeutics where you're targeting that peptide. And there's additionally a peptide-HLA targeting benchmark in the soluble TCR realm that I'll talk about on the next slide. However, TCR-based therapeutics tend to have poor stability and expression, as evolutionarily they have not been evolved for soluble expression. Regardless of the modality, the way that these work is recapitulating the native interaction between TCR and peptide-HLA between a cognate T cell and its target cell, which leads to T cell activation and target cell killing. CD3-based bispecifics, or T cell engagers, recapitulate this interaction by interacting with CD3 and also the peptide-HLA and bridging these two cells together between bystander T cell and target cell, leading to that same T cell activation and target cell killing. This therapeutic approach was validated in 2022 with the approval of a TCR-based CD3 bispecific, Kimmtrak or tebentafusp by Immunocore, which outperformed the standard of care in uveal melanoma. It also opened up an interesting engineering approach, wherein natural TCR affinities in the range of 1 to hundreds of micromolar are insufficient for activity in clinically validated formats. However, if you affinity mature these TCRs to very high affinities down below 1 nanomolar, you start seeing both activity in functional formats as well as clinical efficacy. Because of this, we felt new technologies were required to rapidly discover and engineer TCRs, as well as TCR mimetic antibodies, to generate the next generation of potent and specific peptide-HLA targeting therapeutics. So going into our platform design and validation for this purpose. So our platform for TCR engineering is based on the native CD3 TCR complex and taking the extracellular domains of both the alpha and beta chains from that CD3 complex, importing them into a non-natural bivalent format that we call a TCR-FC. This has engineered constant domains for added stability, as well as an IgG1 FC to port seamlessly into our existing antibody-based platform. As I mentioned, this can be then ported into our engineered yeast for both surface expression, where we can generate variant libraries and do selections for things like higher affinity or higher specificity, or without the need for reformatting, we can go into soluble secretion of these TCR molecules for high throughput purification, binding characterizations, and developability characterizations. To validate this, the platform actually works, we started out with 44 literature-controlled TCRs ranging across 5 different targets and different affinity ranges, from native affinities in the micromolar range all the way down to highly engineered affinities in the picomolar range. These also span diverse alpha and beta variable genes to make sure that this expression was generalizable across the diversity of TCR space. When we actually look at these TCRs on the surface of cells, what we can see is that the expression here on the y-axis and antigen binding to tetrameric antigen on the x showed high binding for the vast majority of our TCRs. In fact, 85% of our TCRs were able to bind to the peptide-HLA antigens above background, suggesting that they were successfully expressed on the surface of yeast. Furthermore, when we solubly secrete these TCRs and purify them through high-throughput purification methods and measure their affinities in a monovalent orientation by an SPR-based carteric kinetics assay, we see that there is a good correlation between the reported affinities and their measured affinities, suggesting that yeast expression was successfully able to recapitulate the native binding properties of these TCRs. With that in mind, we then wanted to see whether we were able to affinity mature TCRs to generate potent and specific therapeutics. For this case study, we chose a model TCR 1G4, which binds naturally to the HLA-A2 NY-ESO peptide. This is a cancer testes antigen that's well studied in the literature. To affinity mature this TCR, we diversified all 6 CDRs of the TCR, which you can see across both the alpha chain and beta chain in the structural representation, contact both the peptide and the HLA, with the CDR3 loops in the center of this structure being the primary determinant for both specificity and peptide contacts. To affinity mature these TCRs, or rather this TCR, we generated a library where each CDR was diversified individually with 1 mutation per CDR. This is a deep mutational scanning approach that we then built into libraries and selected for the highest affinity among the binders. We're then able to sequence out those library selections and compare to the pre-selection libraries in order to generate enrichment ratios for each of these mutations along the CDRs, and then visualize those mutations to then rationally combine them in a second cycle in order to guarantee that we are sampling the highest possible affinity combinations for this TCR. So how did this actually work? Well, again, looking at these expression plots with expression on the y-axis and antigen binding to tetrameric antigen on the x-axis, we can see as we have a strong binding population from the library at the first round of diversification. Moving into the second round, we're then able to pressure for binders that are higher affinity than the parent molecule. The parent is overlaid here in black, and we're selecting for this leading edge of rare clones that have higher expression, or rather higher binding than the parent molecule. And then going into lower concentrations of the tetrameric antigen, we're able to now see that that sliver has expanded, and we're enriching binders that are better than the parent molecule and therefore higher affinity. With this successful completion of these selections, we then moved into next generation sequencing of these libraries and selections to determine our enrichment ratios. What we saw when we did this next generation sequencing analysis was that we had significant enrichments at 5 of the 6 CDRs, so the CDRs 2 alpha, 3 alpha, 1 beta, and actually all of the CDRs within the beta chain. We also had multiple positions within each CDR that had enrichments and some that had multiple amino acids at each position that were significantly enriched. We then were able to take these 36 mutations and rationally combine them into a second cycle library, wherein we were able to sample up to 3 mutations per CDR and up to 12 mutations per clone in a single library. These were built into that single library by transforming both the diversified alpha chains and diversified beta chains into that singular library. Moving into that second round of selections, we were able to see strong binding now, not to tetrameric antigen, but monomeric antigen of that peptide-HLA reagent, where you're already seeing a separation between that library population in red and the parent molecule here in black, suggesting much higher affinity. By round 3, you can clearly see separation between that parent and library, suggesting that these are in fact much higher affinity than that parent molecule. But was that the case? To determine that, we looked at Cartera kinetics again, an SPR-based assay for monovalent affinities, and relative to that parent molecule here, shown as a non-binder, but truly in the double-digit micromolar range of affinities, you can see that we had around a 400,000-fold improvement in the affinity of our progeny from our library, reaching parity with the literature control in this space over 2 rounds of affinity maturation. In addition, we are able to achieve these very high affinity interactions with far fewer mutations, reaching around 34 picomolar with around 10 mutations versus 20 picomolar for a much more highly mutated clone in the literature. In fact, we're able to actually exceed the off-rate threshold around an hour for this interaction from our library, so these clones are actually likely higher affinity than even being able to show here. However, we weren't just interested in affinity. As I mentioned before, we are interested in functional activity for these TCRs to bind and recruit T cells in a bispecific assay, or bispecific format, rather. To test this, we built a CD3 by TCR bispecific, matching the clinical format, and mixed these with CD8 T cells, as well as target cells of interest to assay their ability to kill these target cells and to be activated. What we saw was relative to the parent 1G4 TCR, shown here in red, these high affinity variants of this TCR had over 1,000-fold improved functional activity in both T cell activation and in target cell killing. Furthermore, these elicited a polyfunctional T cell response secreting interferon gamma, TNF alpha, and IL-2, again, about 1,000-fold over that parent molecule. So not only were these TCRs much higher affinity, but also had much higher functional activity in clinically validated formats. However, as I mentioned, we wanted not only potent therapeutics, but we also wanted specific therapeutics. So to test the specificity of these interactions, we went again to an SPR-based assay, here Viacore kinetics, and tested the affinity of our TCR molecules against alanine scan variants of our peptide. So this is taking the original peptide, the NY-ESO peptide, and varying different positions along that peptide to an alanine to look for knockdowns and binding, where knockdowns and binding suggest greater specificity in that interaction. To give you some examples of that, relative to the wild-type TCR, mutating the single position at methionine position 4 in the peptide greatly reduces the binding interaction, suggesting specificity at the position, and mutation of position 5, that tryptophan, entirely ablates binding, suggesting great specificity at that position. When looking across the entire peptide, what you can see is that 5 of the 7 positions mutated here had knockouts for the 1G4 TCR variants that we generated in-house, as well as the literature control in this space, suggesting specificity at each of these positions. In addition, we were able to find that relative to the literature control, our internally optimized TCRs had higher specificity at 2 of these positions, positions 4 and position 6 along the peptide, suggesting greater specificity in general, and we're now working with an academic lab to further characterize the specificity of these molecules and uncover more trends with specificity for high-affinity TCRs. Lastly, as I mentioned, this is a relatively rapid platform, so I wanted to give you a sense of the timeline involved in this project, where this deep mutational scanning-enabled approach with next-generation sequencing took around 5 months to complete. And there are also standard approaches that do not rely on NGS that take around 4 months. So now pivoting beyond affinity maturation, we wanted to see whether or not we are able to discover clones rather than just engineer them. Our motivation for this was that the native immune system or native TCR repertoire for TCRs has some inherent limitations. Particularly in terms of affinity, the native repertoire is highly pruned by thymic selection for clones that are not too high affinity. This is particularly notable in the case of self-antigens, including many tumor-associated antigens, where affinities are highly pruned relative to, say, antipathogen TCRs, where there's not that same need for pruning the repertoire. This protects us from autoimmunity, however, leads to clones that are much lower affinity and harder to find than, say, an antipathogen TCR. In addition, as TCRs are not selected for soluble expression evolutionarily, many TCRs are poor expressing. We saw this from sorting human T cells and cloning them individually into a mammalian expression vector, particularly in CHO cells. And comparing these to traditional antibodies, what we saw was that most of these TCRs had low expression below 100 mg per liter, suggesting that many of these would not be easily expressed in clinically validated formats. However, what we saw was that high yeast expression was linked to high CHO expression. So those expressing well in yeast tend to express well in CHO. So we then came up with this idea of using synthetic yeast libraries to not only get around limitations in the immune system imposed by central tolerance for high affinity TCRs, but also to be able to select for TCRs that had both high affinity and high expression, being able to surpass the native repertoires in these two facets. So how do we actually conduct these library selections? Well, first we need to identify the alpha and beta germlines we'd build into those libraries. So for this, we looked to NGS data sets of the human repertoire for TCRs and identified 20 alpha germlines and 16 beta germlines that each represented around 65 to 70% of their individual repertoires. We also looked at known TCR structure databases and found the same. We then paired each of these germlines with 25,000 human-derived CDR3s per germline in order to guarantee diversity not only in the alpha and beta germlines, but also in the CDR3s, which are responsible for peptide recognition. We then build these into our engineered yeast to be able to sample around 200 billion different combinations of these TCRs. And this is a significant number as it is higher than the diversity in any one human for unique clonotypes, around 10 billion. So really a large diversity space that we're sampling here. However, as those who have worked with yeast before know, you cannot directly sample 200 billion combinations. This is a huge amount of yeast that you would need. So we then break these down into individual sub-libraries, where we take a single alpha variable germline and pair it with all the beta germlines, again with 25,000 combinations of CDR3s per germline, to sample around 100 million unique TCRs per sub-library. However, to get more deeply into that 200 billion combinations, we then reshuffle these libraries. So taking those collapsed diversities from the beta germline and then reshuffling the alpha germline from 1 germline to 20 to generate pools of not only the 16 beta germlines, but also the 20 alpha germlines. Again, with that diversity in the CDR3 beta to generate chain shuffle libraries. And again, sampling around 100 million unique TCRs. We're able to do this not only for the alpha chains, but also for the beta chains as well, single beta by pooled alpha, as TCRs have a unique feature of having equal contributions between the alpha and beta chains in antigen recognition. With this in mind, we then went to 3 model antigens to test our ability to select for binders from these synthetic naive TCR libraries. First, starting with the immunodominant epitope of influenza in the context of HLA-2 presentation. This represents a case where the native repertoire should actually have quite high affinity TCRs, so we're really testing our ability to exceed the native repertoire in both expression and affinity. Moving on to a fine specificity problem with HLA-2 presenting the neoantigen p53R175H. This varies from the native p53 tumor suppressor only at a single position, here at position 8 in the peptide binding groove. So this creates a problem for finding very high affinity, but also very high specificity binders. And lastly, the tumor-associated antigen MART1, and this is a melanoma antigen and a self-antigen where the native repertoire should be highly pruned, so we should be able to find binders that are much higher affinity than the native repertoire. However, to make this more difficult on ourselves, we looked for this rare phenotype of triple cross-reactivity shown in a paper from Andy Sewell's lab around 2 years ago, where a subset of MART1 reactive TCRs also bind these additional 2 tumor-associated antigens, so looking for positive cross-reactivity in this case and a more stringent selection condition. So focusing here on the influenza selections. To show you how these look, I want to show you again these binding plots for 8 different sublibraries that were brought through 2 rounds of Adimab-based enrichment and then followed by a round on the sorter. And you can see from expression and antigen binding to tetrameric antigen that we have a strong binding population after 3 rounds of sorting. In addition, we wanted to check that these binders were specific to our antigen of interest. And to test this, we took a mismatch antigen, this p53 antigen I mentioned on the previous slide, that's entirely non-related to the influenza antigen of interest. And this is to guarantee that our binders are binding in a peptide-centric manner. And what you can see is the vast majority of the library is in fact binding in a peptide-centric manner, not showing binding to the off-target antigen. We're able to clean up the libraries for that peptide-centric binding mode. This is significant because relative to a TCR mimic library, for example, where there's no evolutionary basis for peptide-HLA recognition, usually there's a large portion of binders to nonspecific epitopes. However, with these TCR-based libraries, we're seeing that even though these are synthetic libraries, that peptide-centric binding mode is already enforced natively. So this was very encouraging to see. Finally, in round 5, we're able to move into lower concentrations of this tetrameric antigen, where we're able to sort for the highest affinity binders for both sequencing and characterization. However, as I mentioned before, we were not done at this point. We then reshuffle these libraries to add diversity on both chains, so moving into pooled alpha and pooled beta germline libraries. We again enrich for binders by magnetic enrichment and move on to the sorter here with lower concentrations of that tetrameric antigen, showing that we are in fact improving the affinity of the selection outputs as we go through further rounds. In addition, our previous cleanup against peptide nonspecific binders was successful, as in our shuffle libraries, we do not see any peptide nonspecific binders. So that was encouraging to see as well. Lastly, going into the terminal round of these sorts, what you can see is we're able to drop the concentration of our peptide-HLA antigen down into monomer concentrations, particularly 500 nanomolar monomer, to see a binding population. And this is significant because, as I mentioned before, most TCRs natively have affinities in the micromolar range. So by using this nanomolar concentration of antigen, we're enriching binders that are better than the native repertoire, which we then sequenced and characterized. So how do these outputs actually look? Well, to first test this, we wanted to compare to native repertoires in terms of the binding population sequence diversity. To look at this, we looked at alpha germline representation and beta germline representation in these heat maps. What you can see is that although there are diverse hotspots throughout the alpha and beta germlines, there's this collapse on a single beta germline, TRBV19. And in fact, this is what's seen in natural repertoires of TCRs specific to the same antigen downloaded from the VDJ-DB. You can see that same collapse, and this turns out to be a feature of these TCRs because of germline encoded recognition from this single beta germline that's well studied for this antigen. But it was encouraging to see that our synthetic libraries were able to pull out that same type of interaction. Furthermore, when we drill down on these TRBV19 positive clones, we're able to see features of convergent recognition in the CDR3 beta as well, with lengths that match the natural repertoire from our synthetic libraries, as well as J-region pairings at the tail end of the CDR3 beta that in general match the native repertoires as well. It's only when you really focus in on the central motif of these repertoires that you start noticing some differences, where natural repertoires have this central RS motif that is highly enriched. However, we see that the same motif is not highly enriched in our synthetic libraries. In fact, seems to be outcompeted as you go through further rounds of stringent selections, suggesting that we're able to enrich a more diverse motif, but also potentially a higher affinity motif in these very natural looking TCRs, but that differ in fundamentally important ways. And this was confirmed when we looked at these natural repertoires versus our synthetic repertoires, here looking at monovalent affinities, where by and large, you're seeing comparable affinities in the micromolar range. And this makes sense again for this antigen where they're not heavily pruned by central tolerance. However, we're able to move beyond that natural micromolar affinity into the nanomolar range for our original naive synthetic libraries. And by the shuffle libraries, move into these highly divergent in terms of their affinity clones in the double-digit nanomolar range. So these are very, very high affinity clones, even though they're outlier in this case, just showing the power of the platform to enrich these rare but very high affinity clones. Finally, moving into soluble secretion of these same TCRs, what you can see is that relative to natural repertoires, which tend to have very poor expression, as we've seen over many cases, we're able to enrich a binding population that has very high expression in our yeast-based platform. And we also expect these to have high expression in mammalian vectors based on our previous studies. So we're now expressing TCRs that have much higher affinity as well as much better expressibility. Finally, to show the power of this platform across a diverse set of antigens, just want to show you some binding plots for our two additional model antigens, the p53 R175H neoantigen and that self-antigen MART1. For the p53 neoantigen, what you can see is that, again, by round 3, we have a strong binding population that by and large is showing peptide-centric binding that we're able to clean up. And then moving into round 5, looking at the wild-type antigen, again, differing by only a single amino acid, you can see that we have this large population of selective binders that don't bind to the wild-type antigen, but do bind strongly to the p53 R175H neoantigen. So we're now able to achieve that specificity profile that's desired. In upcoming rounds, we'll be pressuring for the highest affinity of those specific binders. Additionally, for the MART1 antigen, we again have a strong binding population after 3 rounds, here showing much higher amounts of peptide-centric binders as well, with very few clones in this non-specific quadrant. But additionally, we are able to find the desired rare triple cross-reactive population binding to these 2 additional tumor-associated antigens, IMP2 and BST2. Lastly, just because specificity is a very important component of TCR-based therapeutics, as well as peptide-HLA targeting therapeutics in general, I want to show you some of the assays we have lined up for these synthetic-derived TCRs. Particularly, we do have an alanine scan kinetics assay similar to the one I showed you in the affinity maturation pilot, but now adapted to the lower affinity TCRs in an avid orientation. So you can see, again, modifying each of these peptide positions to an alanine to be able to rank order our TCRs for specificity and also compare against natural repertoire TCRs. we can then take those highest affinity TCRs and do a much more thorough testing of their specificity by selecting peptide diversified libraries in yeast, where the HLA and peptide, or rather, the peptide HLA is presented by the yeast and able to select for a specific binding population, showing the diversity of different peptide positions that can be recognized by the TCR. This can then be computationally reconstructed into predictions of off-targets, and each of these off-targets can be validated individually for binding to our TCRs. This allows us, again, to compare against natural repertoires, look for potential off-targets, and then possibly even engineer away from the off-targets during a affinity maturation. So to summarize the presented work, I want to highlight again that our engineered yeasts were able to successfully express soluble TCR Fc proteins that were able to recreate the binding properties of published TCRs in the literature. Additionally, by employing a deep mutational scanning approach, we're able to engineer these TCRs in high throughput for higher affinity and high peptide specificity, facilitating the generation of potent and specific soluble TCR-based therapeutics. Additionally, to overcome limitations in the natural repertoire of TCRs in both affinity and expression, we have generated the first fully human synthetic TCR libraries in yeast, and proof of concept selections of these libraries show that we're able to identify synthetic TCRs that surpass natural TCRs in both their affinity and expression. And lastly, we have additional work ongoing to define the specificity profile of these synthetic library-derived TCRs, as well as generate binders for 2 additional model antigens. And that work will be upcoming and continuing. With that, I want to thank all the many people at Adimab who've contributed significantly to this work, including those in the platform technologies team who have worked on the bench aspects of this project, the computational teams who generated the NGS analysis required for these platforms, the library team who generated these libraries and contributed their expertise to the generation of these, the high throughput expression, protein analytics, core groups, and management teams for their support and their contributions to this work, as well as graphic support, and of course, the folks at Cactus and Scientist.com who hosted this webinar. And with that, I want to thank you all for your attention, and I'll gladly take any questions.
Excellent. Well, thank you, Garrett, for an excellent presentation. So, Garrett, ready to answer a few questions here?
Yeah, of course.
Fantastic. All right. So first one here, Garrett, do you need to modify the glycans in your TCRs to see binding in your platform?
Yeah, that's a good question. There are a lot of glycans in TCRs, and as I mentioned before, these are not typically found in antibodies, so it's a unique challenge for engineering TCRs. But what we found is because TCRs have co-evolved with these glycans there, these tend to not directly modify the binding interface of the TCR, so we don't actually need to modify these in order to see binding, so we can keep everything native in the sequence.
Fantastic, great answer. And so just as a reminder to everyone, if you do have a question that you'd like to ask, be sure to submit it using the Q&A tab. All right, another question here. So does the high affinity of TCRs impact their specificity? And do you see any new off-targets arising?
Yeah, this is a question we get a lot. Just in terms of TCRs, it's thought that by improving the affinity of these, that you're going to start getting off-targets. And this has been true, for example, in cell therapies, where new off-targets arise. But what we found with soluble therapeutics is because it's a binding metric rather than a activation metric, the off-targets at low affinities are also the off-targets at high affinities. It's just kind of shifting the window entirely towards the higher affinity end. So you end up with a kind of specificity window rather than new off-targets. You're just trying to tune the relative affinity between off-targets instead of gaining new off-targets.
Do you check for TCR specificity against viral antigens?
Yeah, not at the moment, but it's an interesting point as the human body will, of course, not only express its own antigens, but also the antigens of the pathogens and also commensal either bacteria and antigens that are within the human body. So it's a good point and something that we can certainly continue to look at.
Excellent. All right, another one here. So when shuffling alpha and beta libraries, how do you determine which alpha line to start with in the initial screening?
Yeah, so there's a lot of different choices, of course, that you can make between which of the germ lines to use as kind of your starting point for these sublibraries. And the way we've gone about this is through multiple different metrics. So first, looking at the expression, we want to make sure that we're building these libraries in kind of a high expressing background, but also we want to make sure that these are the most represented germ lines in human repertoires to make sure they're matching as closely as we can to that human repertoire. And lastly, we're looking at diversity in the sequence. So the lengths of the CDRs 1 and 2 within the alpha chain and beta chain differ between different germ lines and also impact the specificity and the properties in general of the TCR binding. So we want to make sure that we have diverse starting points there. So that's just to give you an overview of the main different metrics we look at.
Fantastic. All right. Well, that's all the questions that we have. So unless any other ones come in, Garrett, thanks so much for your insights today. It's been a pleasure having you with us.
Thanks, Liam. And thanks, everyone, for joining.
Definitely. Big thanks to everyone for joining the webinar today. And in closing, we hope you enjoyed the Scientist.com and Cactus webinar, and we'll see you again next time. Have a great day, everyone.